Hi, once again, welcome to my channel. As I promised, I'll be taking you through more essays. We did from question 1 up to question 6 in our part 1 series. Now I'm here with part 2, and we want us to pick from question number 7. We are looking at form 1 questions that come in form of essay. So we did question 1 to question 6, and remember to watch that video. It's still here. I'll share it in the comment section. You can see it in the prescription. The link is there. If you missed part 1, it is there. We discussed 6 questions that come from form 1 in details. Now, allow me to take you through more questions from form 1 that come in essay form. Remember to subscribe, remember to follow, remember to share, remember to comment so that we can reach as many people as possible. Otherwise, let us continue from where we left. Now, question number 7. Explain why the following conditions are necessary for photosynthesis. Explain why the following conditions are necessary for photosynthesis. And the conditions are 1. Carbon 4 oxide. Carbon 4 oxide. Why is carbon 4 oxide necessary for photosynthesis? Carbon 4 oxide is required in the dark stage of photosynthesis. Now, photosynthesis occurs in two phases or stages. We have light stage, which we call it the light-dependent stage. This is where now the water molecules shall be broken down by light. That's why we call it the light-dependent. Water molecules are broken down by light to form the 4 hydrogen ions and the oxygen gas. So the oxygen gas shall be released to the air, but the 4 hydrogen ions will now proceed to the next stage, which we call the dark stage, or what we call light independent stage. Now, when we go to the light independent stage, this is where now we meet the 4 hydrogen that came from the first or the light dependent stage, now fusing with carbon 4 oxide. And that's why some books call that stage as carbon 4 oxide fixation. This is where now carbon 4 oxide fuses with the 4 hydrogen ions now to give you the food, or what you call the glucose produced at the end of that. So they're saying it combines with the hydrogen ions from the light stage to form glucose, protein, and lipids. So low concentration reduces the rate of production of energy and food, while high concentrations lead to an increase in the amount of energy and food formed. Number 2 is light. Now, I've just talked about light in the first phase of photosynthesis as one of very important, because it helps us to split the water molecule. Remember, the water molecule from the formula, the chemical formula of water is H2O. So it has the hydrogens and then it has oxygen. So light will help us to subdivide or break down the two so that we separate the hydrogens and the oxygens. So it is used to break down water molecules through photolysis into hydrogen ions and oxygen and energy. So the energy and hydrogen ions formed are used in the dark stage. We have chlorophyll. Now, chlorophyll is the green pigment that is found in plants. And this one is majorly to trap light energy from the sun that is used in photolysis of water molecules. Sustainable temperature and pH. Now, whenever you see we talk about pH in any reaction, pH and temperature, that means that reaction or that process is enzyme controlled. That's why you hear the issues of temperature and pH. So for this case, temperature affects the enzymes' activities in the photosynthesis. And therefore, optimum temperature will activate the enzymes. But for maximum production of food, while extreme low temperatures will inactivate enzymes, leading to less or no production of food. But high temperatures, again, denatures enzymes. And this one will do what? will stop the process of photosynthesis. So photosynthetic enzymes work well in low pH. Water. Now, water forms a medium for chemical reaction. It's also split to give yield to hydrogen ions, I had described earlier, hydrogen ions, oxygen, and energy for use in the dark stage. Solvent for material used in photosynthesis. Water is the major solvents that we have, especially in such kind of processes. What is digestion? Now, digestion occurs in two ways. It's either mechanical and chemical. Mechanical, breaking down physically by use of teeth. Chemical, enzymes activities, for example, the ptyalin in the mouth acting on the starch. So this is the two processes, where both the mechanical and chemical will now bring about the effect of, will now bring something called digestion. So it's the process by which the large complex food molecules are broken down into soluble molecules for easier absorption across the intestine or the intestinal wall to blood stream. Now, what is the importance of a balanced diet in human nutrition? What is the importance of a balanced diet in a human nutrition? A balanced diet consists of all types of food types in their right proportions. And this include proteins, carbohydrates, minerals, lipids, water, vitamins, and phages. So, proteins are used for growth and repair of worn out tissues, which also provide energy in the case of acute shortage of carbohydrates or when you're undergoing what you call an extreme starvation. They also bind and transport specific molecules from one part of a body to another. Structurally, proteins support tissues in the body, e.g., the bones, the bone and their cartilages, tissues. They also act as a metabolic regulators, such as the enzymes and hormones. Those are proteins. Then carbohydrates, they are used to produce energy. That is, they have what we call stored energy. Lipids, they produce energy, form of storage of energy. They also insulate the body because they form the layer beneath the skin is actually in lipid form, that is fats and oils, and major structure components of the cell membrane. And when oxidized, it provides metabolic water. Vitamins, they defend against infection, form coenzymes which activate enzymes. Water is a universal solvent. That's a medium for chemical reactions. And it's also used as a coolant because of its high specific heat capacity. It also maintains the shape of the cell, hydrolysis of many substances, transport medium in the body. That's a transport medium in the body. Mineral salts, these are, they aid in the formation of teeth and bones, formation of hormones, e.g. thyroxine, formation of blood, maintenance of osmotic pressure of body fluid, transmission of nerve impulses, Phages, they make food to be bulk, promotes peristalsis and absorption of water in the large intestine. They also induces mucus production. Explain the factors that affect enzyme activities. And these factors are, number one is temperature. Temperature affects the enzyme's activity in which way? Enzymes are protein in nature, and therefore they are very sensitive to temperature changes. As temperature increases, enzyme activity also increases until it reaches what we call the optimum or what we call maximum. Above the maximum, the reaction decreases sharply And this is due to the destruction of the enzyme structure, which become denatured. And this will make the enzyme to be ineffective or non-functional. Now, most enzymes have an optimum temperature of between 35, that is between 30 to 35 degrees and 40 degrees Celsius. Now, when the temperature decreases, the rate of enzyme reaction also decreases as the enzymes become what? Inactive. The enzymes become inactive. pH or acidity or alkalinity. This is what we call the pH, the acidity or alkalinity. How does it affect? Now, most enzymes have optimum pH of close to 7. pH 7 is what we call the neutral, which is what we call the intracellular pH. However, some enzymes work best in an alkaline medium, while others work best in an acidic medium condition. So as the pH exceeds optimum, we're talking about pH 7, the enzyme activity decreases. So extreme acidity or extreme alkalinity denatures most of the enzymes. Substrate concentration and enzyme concentration. Now, enzymes reaction increases with increase in the substrate concentration. We talked about in Form 1 that enzymes are substrate specific, and also enzymes depend on the amount of substrate given. And therefore, if you increase the amount of substrate, the enzyme reaction will also increase. But it won't go for long. It will increase up to a certain level where further increase in substrate concentration does not increase the rate of enzyme reaction. Why? Now, we have been increasing the substrate concentration, but we have not been increasing the enzyme concentration. And therefore, the substrate concentration is increased and all the active sites of the enzymes are occupied. And because they are occupied, the reaction will not be there. However, when the enzyme molecules are increased, there is a proportional increase in the maximum rate of enzyme reaction. So enzymes are, however, required in small amounts, and hence they speed up the rate of biochemical reactions without altering the equilibrium. And this is because the enzymes do not take part. Remember, the enzymes only fastens the reactions or initiates the reactions, but they do not take part in those reactions. We also have what we call enzyme cofactors and coenzymes. Enzyme cofactors and coenzymes. So these are non-proteinous substances which activate enzymes. Now, most enzymes will not work without them. Examples of cofactors are metallic ions such as iron, magnesium, zinc, copper, and also vitamins as enzyme coenzymes, and also vitamins which act as enzyme coenzymes. Now, these substances are required in very small amounts and are used repeatedly and can be recycled. Enzyme inhibitors. Now, these are substances that inhibit, from the word inhibit, that is enzyme inhibitors, they inhibit. These are substances that inhibit enzymes' action by competing with normal substrate for active sites. Now, there are two types. One is known as competitive, and the other one they're known as non-competitive. Now, competitive inhibitors have no permanent effect on the enzyme action, while non-competitive inhibitors combine permanently with the enzyme molecules and thus distorting or blocking the active sites permanently. Now, examples of these inhibitors include cyanides, mercury, silver. which inhibition can be reduced by reducing the concentration of inhibitors, or by increasing the substrate concentration, or by increasing the substrate concentration. So, ladies and gentlemen, that one brings us to the end of Form 1, Part 2 series of the Form 1 essay questions. I'll be back with Form 2, and therefore I beg you to subscribe, share, follow, comment, so that we can reach as many people as possible. Otherwise, for now, I want to say, until we meet in the next video, be blessed. God bless you. Bye-bye.